Welcome, guys. We are going to be doing our notes on antibiotics and isolation versus quarantine today. I'm super excited. So we've been talking about our first part of this unit was all about the different vectors of diseases, so bacteria, viruses, parasites, protozoa, funguses. And now we're starting to talk about, okay, what happens when these things actually enter the human body? How do we as a population, not so much as how our immune system responds, but how do we as a population start to deal with these sicknesses that come on because of these different vectors? So today we're going to be talking about prevention strategies. So some of the prevention strategies we discussed in our lab are things like personal protective equipment like masks, gloves, gowns. That's not something we're going to do a whole lecture on, but it is important to just know that a personal protective equipment is a good way to keep yourself protected from different diseases. We talked about vaccines when we did our lecture, and we're going to do a reading and some questions on it tomorrow. But today we're going to talk specifically about antibiotics and then some separation strategies, which are isolation and quarantine. Okay, so we're doing two separate sets of notes today, but it won't take us very long, and we're going to have a good time learning about how to prevent the spread of diseases. Remember, yesterday we talked all about epidemics, pandemics, and outbreaks. Outbreaks, epidemics, pandemics, in order from least severe to most severe, right? So these kind of strategies come into play when we start to prevent the spread of diseases. So these strategies are going to help us mitigate or lessen the spread of diseases. So let's get started. We are heading to the notes that look like this and this. And you're going to be filling in the different places of the notes with the information on the slides. So our bell ringer, so pause this, or if you're at home, make this. So I would like you to write down epidemic, outbreak, and pandemic on a note card, or pieces of note cards, or just pieces of paper. If you were in class, we already did it. We made small ones. You should have them, but most of you watching this video were not here when we made them. So you can make them. I'd like you to order them on your table from most severe to least severe. Pause this to give yourself a chance to do it, and I'll go over the answers. So we learned yesterday, I'm about to give out some answers, so if you're not ready, pause this. We learned yesterday that the least severe kind of category or rating that we have for the spread of diseases is an outbreak. It's a couple cases, more than we expected, but it's not as many people are affected, and it's not over a large area. The next severe is epidemic. So with an epidemic, to move from an outbreak to an epidemic, you have to have a more concentrated region and more clusters. So you have clusters of people who have the disease. It's spreading more quickly than we thought, and it's spreading to more people in a larger geographic area than if it were just an outbreak. And the very last one that we have is a pandemic. To get from an epidemic to a pandemic, you have a lot more people affected with the disease or coming down with the disease, and over a greater area. In the most severe cases, a pandemic will have cases all over the world like we see with COVID. We saw this, if you remember, I don't know if you were aware of the news, but the coronavirus started as an outbreak. Then it went up to epidemic when it started spreading across Europe and Asia. And then it finally went to a pandemic when it came over here to North and South America. Okay, so now it's all over the world. We do consider it a pandemic, and you have some firsthand knowledge of things that we're learning today. But we're learning about antibiotics right now. So antibiotics, aka antibacterial. So write that down in your notes. Ooh, not what I wanted. So this would be written down right in there. Right here or somewhere, aka antibacterial. They are a medication used to fight infections caused by bacteria. So you can write that down somewhere at the top of your notes. Medications used to fight infections caused by bacteria. Antibiotics, as we learned, will not work on viruses because they only work on bacterial infections. the types, and this is going to go in your little pill bottle of your notes. We have tablets, capsules, liquids, creams, and ointments. All are types of antibacterial treatments or antibiotics. So Neosporin, type of antibiotics. Amoxicillin would be something that you might take if you had strep throat, a bacteria-caused illness. We have some history of antibiotics. We talked a little bit about it the other day, but we're going to hit it one more time just to get it kind of in your head. Antibiotics, and you are going to take this circle and put it in the circle by antibiotics on your notes. This is the cause of death. Make sure you label it. Before 1936, when antibiotics were invented, 70% of deaths were caused, 30% of deaths were caused by bacterial infections. So if they had antibiotics before 1936, they could have easily cured these people that were dying of a bacterial infection. 70% were by other causes, but this advent of the antibiotics is very, very important to our medical and technology history, right? Our advances. So this helps save a lot of people from dying. If we had gotten strep throat back in the day before antibiotics, there's a possibility that we might have died from it, but not anymore, right? Antibiotics will treat most bacterial infections. This history goes down in the scroll-looking thing of your notes. Antibiotics were invented in 1936. We just talked about that. Before that, they didn't have any kind of something that would treat bacterial infections. They kind of just hoped that the people would feel better. They tried to lower their fever, right? Let their immune system fight off their sickness. They would give things that help boost their immune system, like oranges. And also another strategy that they used back in the day was the bleeding. So they would try to bleed you out to get the infection out of your blood, or putting leeches on you. By far, this advent of antibiotics is very important, though, because it almost always will help fight the bacterial infection if taken in time. So penicillin, which is actually a type of mold or fungus, grew in a petri dish. This is how it was invented. This is what you need to write down. Grew in a petri dish. It's a certain type of fungus that naturally produces penicillin. Andrew Fleming found this out or discovered it, right? He had some penicillin growing, and then he noticed that his bacterial colonies did not grow around the penicillin mold, which led him to believe that the penicillin produced some type of bacterial killing something, which we now call penicillin. Some other early antibiotics were produced by bacteria themselves found in ground soil. Today, all antibiotics are produced in a lab. So very important, a little bit of history about your antibiotics. Here's how they work, though. You're going to write this down in your notes. They first attack the cell wall or the coating surrounding the bacteria. Then they interfere with the bacterial's reproduction. Then they block protein production in bacteria. Okay? So they go in, they break down the cell wall right here, this blue line. Then they're going to go in and mess around with the reproductive strategies of the bacteria, so probably the DNA. And then to further make sure the bacteria dies, they're going to interfere with their protein production in bacteria, which means they're going to target the ribosomes. After that, they end up dying because they can no longer reproduce or produce proteins. So the bacteria will end up dying, and then you're okay because the bacteria has died and your immune system can now handle the job. So that's the short on antibiotics. Here is an antibiotic apocalypse. So we have been using antibiotics too much in the current times of our developed countries. Watch this video. It's found on your assignments. It's going to kind of talk about maybe the downfalls of using antibiotics so heavily as we do today. So there are some downfalls. We'll talk about them another day in class. And then lastly, so this is your other set of notes. It's not as much to write down. You're going to talk about the other strategies for prevention, which are isolation and quarantine. You're going to write down the definition in each of those boxes. Blue box goes in isolation, orange box goes in quarantine, and go ahead and color your little pictures there. It'll hopefully help you understand. So isolation, what's the difference between the two? They're both kind of similar, right? We're kind of staying away from people. Isolation happens, though, when you separate a sick individual from the people who are not sick. So in this case, this would be like if you've ever seen the hospitals with the isolation rooms, like the plastic walls. Nobody can go in there without gloves or protective stuff, so they're not taking in anything or getting hurt themselves or getting infected themselves. So that's separating the sick person from those who are not sick, while quarantine separates and restricts the movement of all people, no matter if they're sick or not, who were exposed to the contagious disease to see if they become sick. So the idea of quarantine is that when you are exposed to a person with a contagious disease, you might not be sick, but you stay away from the general population to not get them sick. Okay, you see the difference? So isolation, we absolutely know that somebody is sick, and we move them away from people who are not sick. Quarantine is more of a preventative safety measure. We might have been exposed, so we are going to stay home until we know that we are no longer sick. In the case of COVID, we quarantined for 14 days, 2 weeks, to make sure that we are not sick and we will not spread it to the general population. One of the reasons we do that with COVID, like we said, is because they don't show symptoms till days after they are infected, and you can pass it before you show symptoms. So if you don't even know you're sick, you might feel fine, but you might still be carrying the disease. The very last thing you're going to do today, and for this you don't need to turn anything in, you need to turn in your notes. So the notes that you just took, you need to turn those in. You do not, however, need to turn anything in for this game. This is called Vax. It's a game I learned about not too long ago, but we're going to talk about vaccines tomorrow, but you're going to take what you know about vaccines from our lab on Friday and quarantines and combine the knowledge. We're going to complete the game and see what % of the population survives. So if we go into our assignment on Canvas, here's the antibiotics video I was talking about. Here's the Vax game. This is just a for fun game. Try to see if you can protect the population from the virus. You don't need to do this, but it might help you understand the significance of quarantining vaccines on disease prevention. So this is what the game looks like. You can take the tour if you need to see how to play the game, but here's how we're going to play. We'll click full game. We're going to start on easy. These are my best scores. See if you can beat them. You have 5 vaccines, so you're going to pick some individuals in the population. Each dot stands for an individual, and you're going to vaccinate them. And what happens when you vaccinate them is that you remove a node from the system. See? So I'm removing these people from the system because they can't get the disease and they can't pass it on. Okay, now the red one stands for an outbreak has been detected. Now you're going to click on individuals to quarantine so that the disease doesn't spread to the other population, to the general population. So maybe I want to quarantine this guy because he's near the red one, so I'm going to quarantine him, and our connections decrease. So now maybe I'm going to quarantine this guy. And now maybe I'm going to quarantine this guy. Okay, the outbreak has run its course now. So these people in red got it. These gray people did not. Did you see how I disconnected the links by quarantining people and by vaccinating people? So you click submit, and then you click submit, and you see how many people were infected of the population. There were 50 people. How many people were quarantined, how many people were vaccinated, and how many people were unaffected. 90% of the population survived. 92% survived. 8% did not make it. You can keep your trying, or you can move on to the next level. The more levels you get, the higher you get, the more connected the people are, the more messy your population looks as far as interactions. So again, each line here is an interaction between people to people. See how well you do. See if maybe this disease prevention strategy could be in your future. And let me know how you do. Goodbye, guys. Have a great day. Don't forget to turn in your assignment.